Hello everyone, and welcome to our online lecture series for plant biology. Today we wrap up our three-parter on how materials get moved around within a plant. Fittingly, we will end our lecture with a focus on how water exits. Of course, that's actually where we also began our discussion, by discussing transpiration. The way in which water will evaporate from the inside of leaves and leave the leaf through the stomata. Now that transpirational pull helped pull water up through the xylem. And in pulling the water up through the xylem, we also discussed other properties like cohesion and adhesion that bind to the walls of the xylem. And that water was being pulled up from our roots, which last time we discussed the way in which roots can filter our water. Structures like the Casparian strip, made of polymers like lignin and suberin, that force water into a symplastic rather than apoplastic method of transport. We also talked about how the water surrounding a cell can influence which direction the water will go. If there's a lot of dissolved materials on the outside of the cell, water will leave the cell because that solution is hypertonic. And in order to balance out the amount of water on each side, water will diffuse across the membrane, also known as osmosis, and actually eventually kill the cell and cause the plant to wilt. In situations where the amount of dissolved stuff is even inside and outside the cell, we have isotonic solutions. Water is in dynamic equilibrium, bouncing in and out at the same time, at the same rate. And then we have the ideal situation for plants. Plants in a hypotonic solution, wherein there's more dissolved stuff inside the cell than outside, so water is going to want to rush in to try to equalize the amount of water, and in doing so, it exerts turgor pressure, pressure against the cell wall by the plasma membrane and the vacuole, helping push things out. Now we also discussed the pressure flow hypothesis that relies on this source-sink relationship between the leaves and the roots, whereby sugar is made in the leaves, trafficked through the phloem, first through companion cells that have those nuclei and are the thinkers, and then the sieve tube cells, the Frankenstein's monsters that are there to lift and move all of the sugar down the column. Now, as that sugar moves down in slowly but surely reducing concentrations, we at the same time get pull of the water up the xylem. The fact that these two vascular structures are adjacent to one another is intentional. Water is helped in being pulled up the vascular column by the presence of a concentration gradient of sucrose in the phloem. And we also last time discussed the beginning of how the water actually entered the water column, or the stele of the plant, the vascular stele, going from the soil into the epidermis of the root, either flowing apoplastically through the cell walls, where it's unfiltered but moving quickly, or symplastically, where it's getting filtered by the plasma membrane of these cells, or that method of being doubly filtered, going through the plasma membrane and then through the vacuole and plasmodesmatas, transcellular flow. Now, in going through the epidermis to the cortex, water encountered the endodermis, where the Casparian strip again, made of suberin and lignin, obligate that water to flow symplastically rather than apoplastically. Once it's been filtered through the endodermis, water then passes through the pericycle into the xylem, and then we get back to our story of it going up and eventually out through those stomata. So the driving force behind all of this water movement has been the inherent energy of water. That's right, water does have some energy. You see this energy in Mother Nature, whether it be a river flowing or the tides of the ocean. You see that water has energy in the same way that a falling object has energy. We classify this energy with this symbol, psi. And psi, or water potential, is how we determine where water is flowing in terms of its overall osmosis energy. This is how we determine whether water will be flowing in or out of a cell. This is how we determine whether solutions are hypertonic or hypotonic. The total potential of water, the total potential energy of water, is determined by 5 different factors. The first is fairly obvious to us. We've encountered this before. It's the solute pressure. The solute pressure is driven by what's dissolved in the water, things like salt. And of course, when you have a lot of salt dissolved in your water, distilled water will want to flow from the area where it's distilled to the area where it's salty to help equalize the amount of water in each of these places. Solutes like salt displace water, therefore reduce the amount of energy that it has. Our next component is also, that's a big eraser, that's why that's going that way. Sorry, y'all. Our next component is a physical pressure. Something like a plasma membrane. Something preventing water from continuing to flow in. So, physical pressures, whether they be membrane or cell wall, can usually prevent water from overfilling a space. Next up, we've got the energy that gravity imparts upon water, a psi G term. Gravity obviously has an impact on the flow of water. It will allow rivers to flow downhill, and it'll also prevent how high water can go in a tree. As we'll discuss today, there is a physical limit of the ability of plants to pull water high up above the ground. Gravity puts a cap on that limit because adhesion and cohesion and transpirational pull can only exert so much pressure. Gravity needs to be overcome, and as it turns out, the height limit of trees is at about the mathematical limit of how high they could get given this drag on water potential in the form of gravitational potential. Now, the next two are a little bit less intuitive. The matrix potential, meaning kind of the stuff surrounding the water. It's a difficult term to explain that I won't exactly keep going on and about. But the last one is humidity, psi V. And humidity basically measures the amount of water in the air as it's not dissolved, because the amount of water in the air will impact the amount of water that's kept in a liquid form within cells. Because humidity always does have a pull or a push on water within the cells. So I don't expect y'all to keep track of much of this. Just know that total water potential is largely determined by our first 3 terms: solute potential, physical potential, and gravity. With solutes pushing water one way or the other, physical pushing water usually out, and gravity help keeping it down. But we can illustrate the usefulness of these terms with these two examples. So in our first example, let me go ahead and make another berry here. We have pure water, so distilled water, on the left in both cases. And on the right side, we have salt water. Now in the case of salt water and pure water being adjacent to one another, if there's a barrier there at first that then gets lifted, the pure water, due to solute pressure, and solute pressure alone, will migrate to the salt water. It's a unidirectional flow. You're going to overall move more water towards the salt water to help equalize the amount of water in these two quote-unquote bins. Now in our second example, we have pure water and a plant cell. The plant cell both has a solute pressure. Now, it might not have salt, but it has some sugars, it has some proteins, it has other things that are dissolved in the cell. But it also has a pressure due to the plasma membrane, a physical pressure due to the plasma membrane and the cell wall. So even though it's in distilled water, and water is going to be rushing in, the fact that there is a physical limitation as to how much water can rush in means that water will also be rushing out, or its entry will be prevented. And this is why solutions are hypotonic in plant cells, because there's always a pressure exerted by the membrane or the cell wall that will put an upper cap as to how much water can come in. It oftentimes, or rather at the end of things, it will negate the solute pressure. So despite the fact that the plant cell has stuff dissolved in it, the physical pressure of those structures will be the exact opposite of that. So if the solute pressure is pulling water in, the physical pressure will be pulling water out, or pushing water out rather. So therefore you'll get this balance. Now, if you were to go on and advance in, say, a plant physiology class, you will spend a fair bit of your time actually calculating each of these terms. So while they may be a little bit beyond the scope of a traditional botany or plant biology class, I want y'all to at least know that this equation exists, because it was one of those things that kind of blindsided me and was a little bit more complicated for me as a graduate student studying water mobility in a plant. So the equation exists. Solutes and membranes usually drive most of the force, and once you start getting in tall trees, gravity has a role. And that's where we'll leave it. We'll continue on in this lecture by doing what we did for our phloem elements, the companion cells and the sieve tube cells, but this time for our xylem, because xylem is going to be transporting our water and minerals through one of two different types of cells, either tracheids. You can thank the Germans for that interesting word with its interesting spelling. A German scientist was indeed the first to name it. And our vessel elements. Now we've mentioned that both of these are dead at functional maturity, meaning that they have no cell within them. They're literally just a cell wall. They've been hollowed out of their internal contents in order to allow for much more water flow through them. In fact, it's been estimated that the fact that these cells are dead rather than alive means that there's about 100,000 times more water flowing through these cells than the living cells in a plant. The living materials in a plant are going to do a lot to block up and slow down that movement. And so by making these giant hollow pipes, plants can transport water much, much quicker. Now, there are two major features that are found in our vessel elements and one in our tracheids. Our tracheids are defined by the presence of pit pairs, which you'll see in this much better drawing between our two different tracheid cells. So here, these on the left are our tracheids, and you see that they have little pit pairs, allowing for water to flow in between cells. You have much larger pits in your vessel elements, so these still have pit pairs, in many cases a fair bit larger, but they also have perforated plates. Versus the end walls that we find in tracheids. The tracheids actually do end in a wall. The fact that there is not an easy way for water to flow from one tracheid to the other than those tiny pit pairs, means that there's a lot more water moving through our vessels. In fact, vessels are about 100 times faster in moving the amount of water, specifically because of those perforated plates at the end of the cells, rather than the end walls. I mean, that makes total sense, right? If every single one of your pipes ended in a straw and then connected to the next pipe adjacent to it by a straw, they would move very slowly compared to something where it ends in another opening to that pipe. Fairly straightforward, but we can also do a lot to control the insides of our pipes. And we'll talk about the risks associated with each of these different type of xylem elements in a second. So another feature that we'll keep in mind when discussing our tracheids is that these were the first structures to develop evolutionarily. Tracheids are older and original. Vessel elements came along much later in our phylogenetic history. They were second, and that they occurred many times, many independent evolutionary events. And we'll talk more about that when we get to our diversity section. Tracheids themselves are a little bit weaker to freeze-thaw cycles. Water freezing and water thawing can oftentimes introduce cavities in tracheids, and a lot of the plants that have tracheids and no vessel elements don't really have an easy way to fix that problem. Once an air bubble forms in these dead cell pipes, because the materials in them are not alive, they just kind of let it persist. And that'll be a major problem that we'll go over. Now, as far as the contents of our xylem, y'all will already be familiar with these. You know that water is the primary ingredient moved in our xylem, but you know that there are also ions and minerals that derive from the dirt. These are our essential nutrients, things like potassium that our roots absorb from the soil and then move up the xylem to the different components of the plant that might need them. And then there are occasional organic compounds. Xylem can move things like sugar because the contents of the xylem are collectively referred to as sap. These contents within the vascular column can be moved by a number of different forces. The first is a source sink pressure flow. Y'all already know this, but we're just kind of synthesizing it all into one place in our discussion of our xylem. You also know that transpirational pull can have a big role on the rate of water movement in the xylem. Third, we have root pressure. If the roots have a whole bunch of water in them and the top of the plant has less, diffusion via the apoplast will begin to occur and water will be pulled up the plant in a similar way to what we see in a paper towel. And then our fourth major force is cohesion tension. So water staying together and wanting to pull itself up the vascular column because it's being pulled out by that transpirational pull. That last one, cohesion tension, introduces one of the major risks that occur within our xylem elements. Breaks in the water column. You can imagine breaks in the water column behaving very much like a chain of humans dangling from a cliff. Each one of them is holding the other's arms, and they can continue to pull themselves up as long as everyone is holding on. The moment one lets go, that entire chain beneath the break will no longer be able to be pulled up. And in the place of the tension in the water column, embolisms can develop. An embolism is a full-blown air bubble. You can also get cavitation to occur. And cavitations can lead to embolisms, but cavitations can also be creation of like water vapor to take up this space and then prevent the water column from actually invading back and allowing water to be absorbed. And so here in this hand-drawn illustration of two different vessel elements, you can see that water is traveling up each of these separately, but once an air bubble, like here, is introduced into one of these vessels, the water now has to travel via pit pair to its neighbor and then continue up. Now, because of these pit pairs, this air bubble will be contained within that single vessel element. And that's the main goal of the pit pairs and the perforated plates. If a break occurs, it only takes over one part of the water column. It does not damage or interrupt the whole system. Think about what happens in your own house. If one of the pipes to your sink breaks, since you only got one pipe, you're out of luck. In this case, there are many, many individual pipelines, many individual tracks of vessel elements, and therefore if one breaks, water can be diverted to its neighbor in order to flow. And so that's really the innovation that we see in our perforated plates, helping maintain this cohesion tension of the entire water column. Now, tracheids typically can't fix embolisms or cavitations. Once an air bubble is introduced, the vast majority of plants with tracheids cannot reintroduce water to fill in that gap, which seems really weird, right? When we need to fill in an air bubble in our water column, we just keep forcing more water through and it'll take care of that air bubble. Plants, because all of these cells are dead and their neighbors are dead, they're not going to be able to force water in the same way. So tracheids have the additional drawback of really not being able to fix these air bubbles unless they're in some of the more advanced angiosperms, which have developed methods through root pressure to force water back in. Instead, plants with tracheids, like many of our conifers, giant trees that only have tracheids have developed something called a torus margo structure, which I think is fairly clever. And so this image from your textbook illustrates how the torus margo structure works in a pit pair. So in a pit pair, you have these two cell walls forming a U. The torus margo structure is a cell wall disc held in place by two tethers. Now, if there were to be an air bubble formed over here, it would push on the torus margo structure to press against the cell wall on the other side. But by pressing against the cell wall on the other side, you actually close off that access. So without this structure, the air bubble could potentially spread to the next door neighbor. But with this additional structure, the air bubble will push the torus margo structure, block off the spread of the air bubble, and keep the air bubble contained to a single tracheid rather than allowing it to spread. And so a lot of these structures in those perforated plates and pit pairs are meant to A, transport water, but B, also prevent the spread of air bubbles. Kind of weird to think about it, to see it that way, but it certainly is the case. Now, vessel elements, vessels, exclamation point, nothing else there matters. Vessels can be fixed. Cells around the vessel elements typically do a better job of, due to their size, they can't not be fixed. But vessel elements typically, the plant cannot afford to not fix it, and therefore they've evolved mechanisms of forcing water back into the vessel element to reconnect the water column. Now, there's another phenomenon, in addition to the cavitation and embolisms that can occur and are bad in the water column, called guttation. Now, when a plant in the daytime has its stomata open and is breathing in the CO2, water is leaving via transpiration through the stomata. But in the nighttime, the stomata are not open, they're closed. And yet water can still be used, or still is needed to be transferring through the xylem, still needs to be moving through the entire xylem structure. This means that it has to escape somewhere. And it'll oftentimes escape through the endpoints of leaves. You may have even seen some of these droplets, guttation droplets, that occur when transpiration isn't occurring. These droplets have the same stuff as your sap, because they are exudations from your xylem. That means that they can have a little bit of sugar and a little bit of those amino acids and the other compounds, the organic compounds that are found in the xylem. But I mention this because it has wider ecological consequences. In 2005, there was a study that linked ingestion of these guttation droplets and the pesticide called neonicotinoids, which happen to be put into the dirt to be absorbed by the roots of the plants, to be taken up through the xylem, and in their transport throughout the plant, wherever they end up, they kill pests. So if something is coming by and eating the leaves of a plant, it's going to bite into the sap, and if it's an insect, the neonicotinoid will kill it. But if these neonicotinoids are also being transported through these getacean droplets, that can be bad for some of the insects that drink those getacean droplets. You might see where I'm going with this. One of the insects that does not eat leaves but can derive some energy from drinking these getacean droplets are bees. And the use of neonicotinoids has been associated with colony collapse disorder. So there are a lot of reasons for colony collapse disorder, not all of them figured out, but the use of neonicotinoids fell off sharply as the bees that we were trying to protect by using pesticides that weren't sprayed but rather put into the dirt were then finding their ways to these droplets that bees would drink up, particularly in the morning time, and then be dead 10 minutes later. These getacean droplets can actually have much more concentration of the pesticide than we get in the leaves, and as is chomped down by some of those herbivore pests, therefore causing the bees to die quite quickly. So there's a side effect of a pesticide that was very, very unintended. Traveling through the sap of the xylem, meant to stay in the leaves, but then exuded because plants need to excrete some of that water, not via transpiration, but via getacean in concentrated water droplets. While that's kind of a bummer in terms of one of the findings of our sap, I have another plant sap story that might be a more cheery one. And that, of course, is the very tasty sap story of maple trees. Most commonly the sugar maple, but you can tap other maple trees as well. And so every springtime, during the first major melts of the snow, oftentimes around here in late March, and here you see a map of the range where maple syrups are commonly made, and right at the heart of this range, can't leave the UP out, we have our wonderful state of Michigan. This region produces a lot of maple syrup. Maple trees, during the snowmelt, will begin to traffic water up the tree trunk. In so doing, they're also going to start to move a lot of sugar that's been stored underground up the tree, so that way the buds of the branches have all the sugar necessary in order to make their leaves. As you know, the leafing out of a tree is incredibly quick. Every spring, I'm startled by how fast it happens. The snow melts, there's a few days of the leaves starting to bud out from the buds, and then within a matter of 2 days, it goes from a canopy that you can see through to completely blocked out with photosynthetic leaves. The sugar that's stored in the roots is very helpful in driving this process because otherwise the sugar would have to come from the leaves themselves. But there is a boatload of sugar in there, more than the plant can use in some cases. So therefore, it does not harm the tree long term to take some of its sap from it. Now, we commonly use the sugar maple in order to make our maple syrup, but other trees can be used as well, other maples. We prefer using the sugar maple because it has the highest concentration of sucrose in its xylem sap. Now, the reason why you want a high concentration of sucrose is so that way you don't have to pull out that much volume of sap compared to how much maple syrup you're going to make. If you use something other than a sugar maple, what that means is that you, A, might get a little bit of a different flavor because the other compounds, the organic compounds in the sap can be different. But B, if there's less sugar in there, you're going to need to pull out a lot more water in order to have the same sweetness profile of maple syrup. So we use sugar maples simply because of the fact that they have a higher amount of sugar. Other maples and even walnut trees will work for tapping, but you need to pull out a lot more water to get the same quantity of sugar. And maple syrups are quite beautiful. You will see a seasonal difference in the color patterns of maple syrups. With the early snow melts, the maple syrups are typically lighter, like that which we see on the left side here. And then with the later spring, the maple syrups can be darker. This is because of the amount of water and metabolites being trafficked. Once you get later in the season, you get a little bit less water and a lot more other metabolites being produced by the plant, and therefore the concentration here of other stuff to sugar increases, causing the maple syrup to be a bit darker in color. Now, maple syrup gets its unique mapley flavor from those other organic compounds. In fact, right now, we don't understand the exact medley of things that give maple syrup its very particular maple taste. But if you've had syrup from other trees, like walnut trees or beech trees, you'll note that they do taste different. They don't have that same pleasant smoothness to them that you get with a maple. So, one bummer story about guttation and its ability to poison insects, and one hopefully uplifting story that has you craving pancakes, like I certainly am. Obviously, plant sap and xylem play a huge role not only in how we grow our crops, but even in how we make them sweeter. The last little bit about xylem that I want to talk about before getting to the coup de grace of our lecture, or the way in which water leaves through the stomata, is the fact that these xylem elements are old. Here we have a fossil of a xylem from just over 400 million years ago. And while it's fairly faint, you can tell that this is a complete xylem. First off, because it's connected, and second, because you can see those annular rings that are so characteristic of our tracheids. Hopefully at some point in this class, you'll have the opportunity to visualize tracheids underneath the microscope by your own squash prep. You can separate those rings and create a springy-like appearance to the tracheid that we can see in fossils as old as 400 million years ago. Now, it just so happens that there was a large switch in the atmosphere. Carbon dioxide, or CO2, was incredibly abundant before plants came around. But once we started getting a lot of oxygen production, CO2 became much, much more scarce, and oxygen entered the atmosphere. And because oxygen entered the atmosphere, plants ended up losing a lot more water in order to get the same amount of CO2. There have been some studies that estimate that it took about 17 times more water loss in our current ambient oxygen levels as compared to the amount of oxygen loss if you looked at the amount of CO2 prior to the Carboniferous, which is kind of mind-blowing that once oxygen got in the air, plants had to lose more water in order to get the same amount of CO2. Once they started to lose more water, they evolved xylem. And once they evolved xylem, they then started growing taller. Whoa, evolution is pretty cool. Now, the last component of our lecture is, of course, the way in which water leaves through the leaf via the stomata. We know that stomata can operate as open cells with each of the two guard cells resembling a crescent shape, or they can be closed. And when the cells are closed, they have water that's rushed out. Meaning that the plant is wilted. The stomata are wilted. So it's kind of weird to me that stomata have a rectangular shape when wilted and a crescent shape when pushed full of water in a hypotonic environment. How does that work? Well, it has to do with the shape of their cell wall. Stomata have cell walls built in a different way than all of the other cells in a plant. That different construction of the cell wall is called radial. Ooh, let's try to write an R, huh? Radial myceliation. Which basically means that the cellulose fibers of the cell wall are not laid out in the typical fashion. They're laid out more like rays from the sun. Let's illustrate this here. They are radially mycelated, so they have regular spaces, mycelations, coming from the central pore. What this means is that when these two cells, kind of like balloons, are not fully inflated, they end up closing the space. As you start inflating them more and more, due to things like structures of the cell wall operating like pieces of tape on the outside of these balloons, they bend when they're fully inflated rather than staying straight, creating that pore that is the stomata. So that's the radial mycelation, the tape on the outside of a balloon that allows stomatal guard cells to be a crescent when open and a rectangle when closed. You will notice that these guard cells also have chloroplasts that they use to detect sunlight, because guard cells are highly regulated structures. You want to have the guard cells open when you need to perform photosynthesis. Ran out of space there. And closed when it's dark out or when you are short on water. Right? Because if the plant is running out of water, you want to stop losing it through these stomata because it's being pulled out via transpiration. So therefore, in order to optimize the opening and closing of stomata, plants have evolved a very cool regulatory pathway for this. This regulatory pathway revolves around the concentration levels of two different compounds. Potassium in the morning and sucrose or sugar in the afternoon, evening. So as you should with any graph, there's got to be axes labels. On the x-axis, you have time. On the y-axis, referring to our black line, you have the size of the stomata opening, meaning this size, the size of the pore. And on the other side, you have the concentration of the substance. This referring to either sucrose or potassium. And so, in the morning, before the plant has ramped up photosynthesis, the chloroplasts in the stomata are able to detect the first presence of sunlight. They need to open up very quickly to make sure that the CO2 is ready and available once enough sunlight energy has accumulated, which literally takes a matter of a minute in most cases. In order to rapidly go from closed to open, the plasma membrane of these guard cells will start selectively pumping in a whole bunch of potassium. Now, why does a whole bunch of potassium help open up and fill the stomatal guard cells with water? Well, going back to our discussion of hypotonic, hypertonic, you know that when there's a whole bunch of solution or solutes, like salts dissolved inside the cell, that means there's a whole bunch less water compared to the surrounding. So if a whole bunch of K plus comes in, a whole bunch of water will flow in after it to try to equalize the amount of stuff dissolved inside. So the influx of potassium actually causes water to follow along for free. Kind of cool that since you can't regulate where you're pumping the water, you can regulate where you're pumping the ions, and the ions will follow where the water will follow where the ions go. And so there's a rapid influx of potassium that causes the stomatal opening to increase. Now the stomatal opening keeps getting bigger and bigger and stays at its maximum size up until sometime around the midday. In the midday, the plant has accumulated a whole bunch of sugar, and it keeps making a whole bunch of sucrose. And as sucrose reaches its pinnacle during maximum photosynthetic time in the day, because, sucrose is the byproduct of photosynthesis, it begins to get trafficked down to the roots. As the sucrose is being trafficked down, it's decreasing in its concentration. So once a plant goes from peak to decreasing levels of sucrose, we see that the stomata is picking up on this signal that we aren't maintaining our extremely high levels of sugar, and therefore it's probably getting to nighttime because we're not as photosynthetically active, and therefore we should close our stomata. It's super clever, right? At least I think so. So we have potassium opening the stomata in the morning and decreasing concentrations of sucrose closing the stomata at night. There are other environmental cues that can have an impact on this daily opening and closing, because this potassium sucrose model is what's basically operating every single day. Stomata can close in response to high temperatures, like when it gets too hot and plants are losing too much water, the stomata will close, again with wilting. Or if it's getting too cold, stomata will close. Humidity. Some plants will respond to extremely dry conditions by closing up their stomata, as well as air currents, which are another way of measuring humidity, because when air is moving very quickly, things are less humid, You lose a lot more water, and therefore stomata will close. But there are also hormones that can regulate the closing of stomata, like abscisic acid, which is a signal for leaf stress. Something's happened, I'm stressed out, the whole leaf is stressed out. You know what? It'd probably be a good idea to hold on to your water, so let's go ahead and do that. And so you have these environmental cues and hormonal cues that can influence your ability to open your stomata beyond just potassium and sucrose. We also have the same graph derived from your book with real data, so that way y'all can see how this looks with some true real data, with both potassium staining and sucrose staining, and all of the units associated with them. And so that's it. The water will leave through the open stomata in order for CO2 to get in. As the water was leaving, it was pulling up a lot of the nutrients, and in some cases, some of the sugars necessary to help the plant grow. The water was carried up through a variety of forces that have to do with the structure of those dead cells, those xylem that help bring it up. And all of this, all three of these last lectures, connecting from our roots and the nutrients and water that they absorb to the leaves and the stomata by which water leaves the leaves, hopefully paint a whole picture of this entire process and lifespan of water within a plant. So I will end this series of lectures by sharing one of my favorite questions I was ever asked by a professor. We were having a discussion, and this professor liked to personify things. It always helped people remember things better when they felt they were going through it. So this professor asked me to pretend that I was a molecule of water, just dripped from the sky in a drop of rain, near the roots of a thirsty plant. Tell me everything about my journey through the plant. I encourage you to take on that discussion in studying this material, because in all likelihood, there's a good chance that you might get something similar on the assessments in our class. If not the assessments in our class, then our discussions. I'll see y'all next time when we leave our discussions of the water column and instead explore this last thing that we talked about in stomatal regulation. Plant hormones. Because yeah, plants have hormones too.